to start. So in today's topic, we'll be discussing about the locomotor structures, cilia and flagella in the cell. So how does their structure look? So the structure of ultrastructure of cilia and flagella will be studying about. So they have asked many times the diagram of flagella with that of explanation. So apart from that, even in NEET question papers, they have asked it, and CET question paper also, the labelings they have asked it. Even this year, 2020, they had given a diagram of flagella and asked to find out which connects the peripheral tubules to the central sheath. So when I am describing about that, I'll be informing you about it. Okay. So now I've also shared yesterday's class videos in the classroom, so you can go through that. So the cilia and flagella, so they are the locomotory structures. So they are hair-like outgrowths of the cell membrane. Okay. So cilia, they are small structures which work like oars. See, oar in a boat, you might have seen the boatsmen using the oars. So similar to that, the cilia works. So the movement would be almost similar to oars. So the circular movement of it. Okay. So the small structures which work like oars, they cause the movement of cell or surrounding fluid. So they either help in the movement of the cell or the surrounding fluid they help in moving. So that is the function of cilia. Their movement is similar to oars. So whereas flagella, they are longer in length. They are responsible for cell movement. Majorly, they are involved for the cell movement. Flagella of prokaryotes and eukaryotes are structurally different. You should understand that flagella of prokaryotic cell, and a flagella of eukaryotic cells, they are structurally different. Now, if you look into the structure of the cilia and flagellum, they are covered with plasma membrane. So they have an outermost covering called as membrane. Their core, the central part of it, axoneme, has many microtubules running parallel to the long axis. So the cilia and flagellum are covered by plasma membrane, and you should also realize that their core, the core of a flagella or cilia, which is called as axoneme, has many microtubules running parallel to the long axis. So axoneme, if you look into this core, inside of this core part of it, axoneme, they have 9 pairs of doublets. You can count them. 9 pairs of doublets are there in the peripheral microtubules, we call it as. Towards the side, they have axoneme has 9 pairs of doublets of radially they are arranged peripheral microtubules, and a pair of central microtubules. So they have a 9 pairs of peripheral radially arranged peripheral microtubules and a pair of central microtubules. So this type of arrangement, we call it as 9 plus 2 array. So 9 plus 2 array. So and a pair of central microtubules, they have 9 pairs of radially arranged peripheral microtubules, which are in doublets, and centrally arranged a pair of microtubules. So we call this type of arrangement as 9 plus 2 array. So a pair of, you find this peripheral, radially arranged peripheral, uh, 9 pairs of radially arranged peripheral doublets, and at central axis you find a pair of microtubules. So we call this type of arrangement as 9 plus 2 array. This is called as 9 plus 2 array. So the axoneme, you can notice that. Now the central tubules, the microtubules or the tubules are connected by bridges. So the central tubules are connected by bridges, and they are enclosed by a central sheath. You can see this reddish colored one. This is the central sheet. So the central tubules are connected by bridges and they are enclosed by a central sheath. So it is connected to one of the tubules of each peripheral doublet by a radial spoke. So it is the central sheath is connected to each one of the pair of peripheral doublet by a radial spoke. So this is what they had asked in the CET examination also 2020 CET paper. They had asked about this. So the central sheath is connected to the peripheral doublet by, they had given different option like interdoublet bridge, the plasma membrane, radial spoke, central microtubule. So they had given different option. The right answer is the radial spoke. So radial spoke connects the peripheral doublet to the central sheath. So it is connecting the peripheral doublet to the central sheath. So radial spoke is doing that job. So you should understand about it. So thus there are 9 radial spokes. So you can count them. There are 9 radial spokes and the peripheral doublets are interconnected. See each peripheral doublet, they are interconnected. They are connected to the central sheath by radial spoke and each peripheral doublet, they are interconnected by linkers. So you can see the small green one, which is nothing but linkers. So each peripheral doublet is interconnected by linkers. So the cilium and flagellum, they emerge from centriole-like structure called as the basal bodies. So the cilium and the flagellum are arising from the centriole-like structure called the basal body. So basal body, hook and filament. So that is what we had described the flagella as. So it arises from the basal body, which is centriole-like structure. So both cilium and flagellum, they arise from centriole-like structure called as basal body. Okay, so that is what you have to remember about. So then there is interdoublet bridge or the linker, what we call it, as you can notice that. So these are the diagrams that you have to remember about. So the cilium and flagellum emerge from centriole like structure called as the basal bodies. So let us just have a gist of this entire uh, basal body. The cilium and flagellum, they are hair like outgrowths of the cell membrane. So cilia and flagella are hair-like outgrowths of the cell membrane. Cilia are smaller structures which work like oars of a boat. So the movement is similar to the oar movement. So they cause the movement of the cell or the surrounding fluid. That is the function of cilia. Flagella are longer than cilia. They are responsible for the cell movement. Flagella of prokaryotes and flagella of eukaryotes are structurally different. So the flagella of prokaryotes and eukaryotes are structurally different. So they are hair-like outgrowths. You can see them. And when you take a section of this flagellum, so the cilium and flagellum are covered with plasma membrane. Their core, this within this plasma membrane, the area we call it as their core is called as axoneme. It has many microtubules running parallel to the long axis. So it has many microtubules running parallel to the long axis. Axoneme has 9 pair of radially arranged peripheral microtubules, 9 pair of doublets of radially arranged peripheral microtubules. So the axoneme has 9 pair of doublets peripherally radially arranged peripheral microtubules. So we call it as peripheral microtubules. 9 pair, you can count them. So even when you're drawing diagram, you should see to it. This diagram is important. You have to practice it. So diagram of chloroplast, diagram of cilia and flagella, and diagram of the mitochondria, very important. And fluid mosaic membrane, fluid mosaic model of plasma membrane. That is also an important diagram that you have to practice. I've also told you a few times, they have even asked the ultrastructure of plant cell and ultrastructure of animal cell as a neat label diagram in the examination. So you have to practice all these diagrams and the labeling should be perfect and precise. Okay, so the axoneme has 9 pairs of doublets which are radially arranged called as peripheral microtubules and a pair of central microtubules. So this is called as 9 plus 2 array. This is called as the 9 plus 2 array. So the central tubules are connected by bridges. So interdoublet bridge, we call it as, not the interdoublet bridge, they are called as the bridges, okay, and are enclosed by a central sheath. You can see this reddish colored one, they are the central sheath. So the central tubules are connected by bridges and they are enclosed by a central sheath. It is connected to one of the tubules of each peripheral doublet by means of radial spoke. So the central sheet is connected to the peripheral microtubules, one of the peripheral microtubules by means of radial spoke. So I told you just now that it was asked in the 2020 CET examination. Okay, so the radial spoke. So radial spoke connects the one of the peripheral microtubules to the uh, radial spoke. Okay, so to the central sheath. So we call that as the radial spoke. So radial spoke connects one of the peripheral microtubules to the central sheath, and we call that as radial spoke. So thus, how many radial spokes are there? There are 9 radial spokes which are arranged. So the peripheral doublets are also interconnected by linkers. So you can see this light colored green structure. So the peripheral doublets are also interconnected by linkers. Okay, the cilium and flagellum emerge from the centriole-like structure, which is called as the basal membrane. So the cilium and flagellum are emerging from a centriole-like structure called as basal bodies. So basal bodies, they are arising from. If they ask you draw a neat label diagram of cilia, you have to draw the same diagram. If they ask you neat label diagram of flagella also, you have to draw the same diagram. Okay, so remember about this ultrastructure of cilia and flagellum. Then the ninth uh, cell organelle that we are going to discuss about is the centrosome and centrioles. So these centrosome is an organelle which contains two non-membrane bound cylindrical structures called as centrioles. So centrosome is an organelle which contains two non-membrane bound cylindrical structure. They don't have any membrane bound. So non-membrane bound cylindrical structures called as centrioles, which are arranged perpendicular to each other. So these centrioles are surrounded by pericentriolar materials. So the centrioles are surrounded by the pericentriolar materials. So the centrioles lie perpendicular to each other, and they are made up of, so what are these centrioles made up of? They are made up of 9 evenly spaced, so you can see this, 9 evenly spaced peripheral fibrils of the tubulin. So 9 evenly spaced peripheral fibrils of tubulin, you can notice. So the centrioles lies perpendicular to each other. They are made up of, each centriole is made up of 9 evenly spaced peripheral fibrils of tubulin. So we call this peripheral fibrils of tubulin. So each of the peripheral fibril is a triplet. You can find three microtubules here. Each of the peripheral fibril is a triplet. The adjacent triplets are also linked. That is the neighboring. ಅಕ್ಕಪಕ್ಕದಲ್ಲಿರುವಂತ the triplets are also linked. So each of this peripheral fibril is a triplet. The adjacent triplets are also linked by this linkers. So you can notice this centriole structures. Okay, so the centrosome is an organelle which contains two non-membrane bound cylindrical structures called as centrioles. See, centrosomes are found only in animal cell. They are absent in plant cell. It's a very important differentiating feature between plant cell and animal cell, especially during cell division. These centrioles, they are going to form the asters. So asters from which the spindle fibers arise. But in case of plant cell, there are no asters. So you have to remember about that. They do not have any asters or spindle fibers because the centrosomes are absent. So that is one thing that you have to remember about. So let us understand this part of it. So centrosome is an uh, organelle which contains two non-membrane bound cylindrical structures, which we call it as centrioles. Okay, they are surrounded by pericentriolar materials. So the centrioles are surrounded by pericentriolar materials. The centrioles lie perpendicular to each other. They are made up of 9 evenly spaced, so they are all evenly spaced peripheral fibrils of tubulin. So each of the peripheral fibrin is a triplet. They have three microtubules. So each of the peripheral fibril is a triplet. The adjacent triplets are also linked by this linkers. Okay. So the central part of the centriole is also proteinaceous. So the central part of the centriole is also proteinaceous and it is called as the hub. So the central part of the centriole is called as the hub, which is connected with tubules of the peripheral triplets. So the central part of the centriole is also called as hub, which is connected to the peripheral tubules of the peripheral triplets, each of which is a triplet. You know that peripheral triplets by a radial spoke made of protein. The central part of the centriole is also proteinaceous and we call it as the hub. So the hub is connected with the tubules of the peripheral triplet by a radial spoke, similar to the central sheath being connected with the peripheral microtubules of the flagella by this radial spoke. Similarly, even here, the hub is connected to the peripheral triplets by radial spokes. This radial spoke is also made up of protein. The centrioles form the basal body of cilia or flagella. That is also one of the important aspect of this. Centrioles, they form the basal body of the cilia or flagella. So I have already told you the basal body is made up of centrosomes. So the centrioles form the basal body of the cilia or flagella and spindle fibers that give rise to spindle apparatus. So they form the asters during the animal cell division. So and spindle fibers that give rise to spindle apparatus during cell division in animal cells is also brought about by this centrioles. So the centrioles form the basal body of the cilia or flagella, and the centrioles also form the spindle fibers that give rise to the spindle apparatus during cell division in animal cells. So this is seen only in case of cell division in animal cells, where the centrioles, they are going to form the spindle fibers. So we call the centrioles which moves towards the opposite poles as the asters. Okay, and they form the spindle fibers that give rise to the spindle apparatus during animal cell division. So that is one point that you have to remember regarding the centrosomes and centrioles. Centrosomes are found only in the animal cells. Centrosomes are absent in the plant cell. The next part of it that we will be discussing about important cell organelle is the nucleus. So an important cell organelle that we will be discussing about is the nucleus. So nucleus was first described by Robert Brown. So the nucleus was first described by Robert Brown. Okay, the material of the nucleus stained by the basic dye was given by the name chromatin by Fleming. So within the nucleus, the material that was stained by the basic dyes or alkaline dyes was given the name chromatin by Fleming. So Fleming terms the name chromatin whenever the material within this nucleus which was stained by this alkaline dye or basic dye was given the name chromatin by Fleming. So nucleus was first described by Robert Brown. So the chromatin was first termed by nomenclature was used by Fleming. He named the term chromatin. So the within the nucleus, the material which was stained by the basic dye was named as chromatin by Fleming. So normally a cell has only one nucleus. Some of the cells, like if you look into paramecium, they have macronucleus, micronucleus, they are binucleate. Some of the cells have more than one nucleates. Some mature cells, they lack nucleus. So mature cells are, a few of them are lacking nucleus. They are like mammalian RBC, except for camel. All the mammals are enucleate RBC. Enucleate means nucleus is absent. So in RBC, you don't find nucleus. And sieve tube cell of plants, vascular plants, the only living plant cell without nucleus is sieve tube. So the phloem component is the sieve tube. The sieve tube cells of the vascular plants, they do not have nucleus. So you should remember some of the mature cells like mammalian RBC, except camel. So camel, which is the ship of the desert. So the camel's RBC has nucleus. Except for camel, all the mammals will have anucleate or enucleate RBC. So RBC is without nucleus. And in case of angiosperms, the sieve tube is without nucleus. So the sieve tube cells of vascular plants are without nucleus. So these are the things that you have to remember about the uh, introduction of the nucleus. So nucleus was first described by Robert Brown. So the material of the nucleus, which was stained by alkaline dye or basic dye, was the stained material was given the name chromatin by Fleming. Normally, a cell has only one nucleus, but there are cells which has more than one nucleus. Like in case of paramecium, it is binucleate. I told you macronucleus, micronucleus. You can find. Okay, so some of the mature cells they lack nucleus. Example is mammalian RBC, except camel. Okay, and sieve tube cells of vascular plants they also lack nucleus. So they are all enucleate cells. So nucleus, the interface nucleus. If you study the nucleus, the best stage to study is the interface nucleus. They contain a nuclear envelope or nuclear membrane. This is in case of a eukaryotic nucleus we are describing about. They contain a nuclear envelope or nuclear membrane. Okay, so they have nuclear matrix, which we also call it as nucleoplasm. So the fluid part or the matrix within the nucleus, we call it as nucleoplasm. And the basic dye-stained material, we call it as chromatin. Then there you find a darkly strange structure called as nucleolus. Many times we call nucleolus as nucleus of the nucleus. See, nucleolus is many times called as nucleus of the nucleus. Nucleus a nucleus adu. Nucleolus. So the presence of nuclear envelope and nucleolus is a characteristic feature of eukaryotic organisms. So they have a nuclear envelope and nucleolus. In case of prokaryotes, nuclear envelope and nucleolus are absent. So within this nuclear envelope, you find numerous opening, which is called as nuclear pore. And you can also notice that the nucleus is connected to the endoplasmic reticulum, so which has ribosomes, that is the rough endoplasmic reticulum and smooth endoplasmic reticulum. So the nuclear envelope is connected to the endoplasmic reticulum. So the with ribosomes, that is rough endoplasmic reticulum and smooth endoplasmic reticulum. So you can notice those features. Nuclear envelope. So when we study about them, it is a double layered membrane with a space between the outer and inner membrane. So nuclear membrane or envelope is double membrane. You can find two membranes here with a space between. So that space is nearly 10 to 50 nanometer. And we call the space between the outer membrane and inner membrane, there is a space which we call it as perinuclear space. And that perinuclear space is 10 to 50 nanometer. Nanometer is 10 to the power of minus 9 meter. So 10 to the power of minus 9 meter is nothing but nanometer. So they have a perinuclear space which is 10 to 50 nanometer in diameter. So that is the space. So we call that space as perinuclear space. So this perinuclear space, the double layered membrane, so it is a barrier between the materials that is available in the nucleus and the cytoplasm. So they act as a barrier. The nuclear envelope, it acts as a barrier for the material present, uh, f- for the material present in between the nucleus and the cytoplasm and vice versa. So the outer membrane usually continues with the endoplasmic reticulum, and they also bear ribosomes on it. So that is the rough endoplasmic reticulum. So the outer membrane continues with the endoplasmic reticulum, which also bear ribosomes on it. So that is nothing but rough endoplasmic reticulum. So nuclear envelope has minute pores. So the nuclear envelope has minute pores formed by the fusion of its two membranes. So the nuclear pore has minute pores. The nuclear membrane or nuclear envelope has minute pores, which is formed by the fusion of these two membranes. These are the passages for the movement of RNA and protein between the nucleus and the cytoplasm. So nuclear pore are the passages for the movement of RNA and protein between the nucleus and the cytoplasm. So nuclear pores are passages formed by the fusion of its two membranes. These are the passage for the movement of RNA and proteins between nucleus and the cytoplasm. That is about nuclear envelope. So chromatin, a network of nucleoproteinaceous fiber. So chromatin is a network of nucleoproteinaceous fibers. So you should remember about that. Chromatin is a network of nucleoproteinaceous fibers. So what does this chromatin consist of? It contains DNA and basic proteins. So chromatin contains DNA and the basic proteins or alkaline proteins. So it is basic in nature. So that alkaline proteins, we call it as histone. So the chromatin is a network of nucleoproteinaceous fibers. Okay, so it contains DNA and the alkaline proteins. Alkaline proteins are called as histones, and they also have non-histone proteins, that is non-alkaline proteins are there, and RNA. So the chromatin, it is a nucleoproteinaceous fiber. It's a network of nucleoproteinaceous fiber. And what is it composed of? It is containing the basic proteins, which we also call it as histones. It contains DNA and basic proteins or alkaline proteins, which we call it as histone. They also have non-alkaline proteins or non-histone proteins they have, and RNA is also present. Majorly, they're composed of DNA and basic proteins, histones. They also have non-histone proteins and RNA. So during the cell division, the chromatins are going to condense to form chromosomes. So that is what is the future fate of this chromatin. During cell division, the chromatins are going to condense to form a structure called as chromosomes. So nucleolus is one or more non-membranous spherical body. So they don't have any membrane. So nucleolus is one, it can be one in number or more than one non-membranous spherical bodies. It is continuous with the nucleoplasm. So the matrix of nucleus, we call it as nucleoplasm. And nucleolus is the site for ribosomal RNA synthesis. rRNA is synthesized in nucleolus. Remember this point for CET and NEET. Ribosomal RNA is synthesized in the nucleolus. So nucleolus is a site for ribosomal RNA synthesis. I've already told you nucleolus is the nucleus of the nucleus. It is a nucleus for the nucleus. Okay, so it is a site for ribosomal RNA synthesis. rRNA is synthesized in the nucleolus. Remember about it. So it is one or more non-membranous spherical body. It is continuous with the nucleoplasm. That is the matrix part of it. See, nucleolus is also a site for ribosomal RNA synthesis. That is one point that you have to remember. It is also a site for ribosomal RNA synthesis. Next, we will be discussing about the chromosomes. Okay, so chromosomes I've already told you during cell division, the chromatin material condenses to form the chromosomes. The best stage to study the chromosome structure is metaphase stage because maximum condensation would have taken place at the metaphase stage. So you can study different types of chromosome at the metaphase stage, or if you want to study the structure of a chromosome, the best stage to study is the metaphase cell division stage. That is the best stage to study the chromosomal structure. A human cell has about 2-meter long DNA distributed among its 46 chromosomes. See, we have 46 chromosomes deployed, 2n equals 46. Haploid is 23. So we can also call it as 23 pairs of chromosomes we have. Of this 23 pairs of chromosomes, the DNA of this 23 pairs of chromosome is nearly equivalent to 2 meter in length. See, DNA is microscopic, but if you are able to join all the DNA, it would be 2 meter long DNA in case of human beings. 46 chromosomes or 23 pairs of chromosomes of human being has 2 meter long DNA. So every chromosome, if you look, has a primary constriction. So this primary constriction, we call it as centromere, on the sides of which disc-shaped structures, kinetochores, are present. So every chromosome has a primary constriction, which we call it as centromere. So centromere is found, the primary constriction is also called as centromere. So 1 mark question they might ask you during the examination, or they can ask the same concept as MCQ question in case of the uh, your NEET as well as CET examination. So every chromosome has a primary constriction called as centromere, on the sides of which you have disc-shaped structures, so which we call it as kinetochores. So on the sides of which the disc-shaped structures are present, so which we call it as kinetochores. So here a typical structure of it, you can notice that it is having two sister chromatids because they are these chromatids of the same chromosome. We call it as sister chromatids. So they are called as sister chromatids, within which you find a pair of chromatin fibers, highly coiled chromatin fibers. And if you uncoil this highly coiled chromatin fibers, you find the nucleosomes, which are, it is just like a bead of chain. Okay, when you uncoil this chromatins, you find this nucleosomes which are wound by this DNA, and two nucleosomes are interconnected by this linker DNA. So that is the nucleosome model. Okay, so they are a bead of chains like, so the chromatin fibers, they are highly coiled in case of this chromosomal arms. When you uncoil them and study them, you find that they have nucleosomes or new bodies. So this nucleosomes are wound by the DNA, and two nucleosomes are connected by this linker DNA. It is like a chain of beads where the thread part is the DNA and the beads are the nucleosomes. Okay, so the uncoiling of this chromatin network, you can do it, but you don't have it for your study, the nucleosome model. Anyway, remember about this sister chromatids. The sister chromatids has a highly coiled pair of chromatin fibers. See, based on the position of the centromere, chromosomes are of four types. Okay, MSAT, you can remember it as MSAT. Okay, based on the position of centromere, the primary constriction we call it as centromere. Chromosomes are of four types: metacentric chromosome, submetacentric chromosome, acrocentric chromosome, telocentric chromosomes. So based on the position of the centromere, the chromosomes are of four types: submetacentric chromosomes, acrocentric chromosome, telocentric chromosomes. So these are the four types of chromosomes that you have. So the metacentric chromosome, you can notice here the metacentric chromosome, the middle centromere forming two equal arms of the chromosome. So this type of position of the centromere is exactly at the center of the chromosome, and they have two equal arms of the chromosome. So this we call it as metacentric chromosome. Submetacentric centromere is a little away from the center. So centromere is nearer to one and forming one shorter arm and one longer arm. So centromere is nearer to one end and it is forming one shorter arm and one longer arm. So this is metacentric, and you can notice here submetacentric. So the centromere is uh, away from the center and it is nearer to one end, forming one shorter arm and one longer arm. Okay, so this sort of L-shaped, see V-shaped, you find it in the metacentric chromosome. Submetacentric, it would almost be L-shaped. So a little away from the center, the centromere is located, and the centromere is nearer to one end, forming the shorter arm, and the opposite is the longer arm. So acrocentric chromosome, you find that centromere is close to its end, forming one short arm and one very long arm. So it is almost very close to the uh, end. So they are subterminal in position. That is also you can write it as. So centromere is close to its end, or it is subterminal in position. Centromere is subterminal in position, or it is close to its end, forming one very short and one very long arm. So in telocentric chromosome, the centromere is terminal in position. So whereas in acrocentric, it is subterminal in position, or centromere is close to its end, forming one very short arm and one very long arm. Okay, so V-shaped, L-shaped, J-shaped, I-shaped. Telocentric chromosome, the centromere is terminal in position. Okay, some chromosomes have non-staining secondary constrictions, so which are called as satellite. So some of the chromosomes are having a non-staining secondary constrictions, which we call it as satellite. Okay, remember about it. Chromosomes with satellite, we call it as SAT chromosomes. Okay, some chromosomes are having non-staining secondary constriction, which we call it as satellite. So it is chromosomes with satellite, we call it as SAT chromosomes. So based on the position of the centromere, the chromosomes are of four types: metacentric chromosome, submetacentric chromosome, acrocentric chromosome, and telocentric chromosomes. In metacentric chromosomes, the centromere is exactly at the center of the chromosome, and they have two equal arms. Okay. In case of submetacentric chromosome, the centromere is away from the center, and it is nearer to one end, forming a short arm and a long arm. In case of acrocentric chromosome, the centromere is subterminal in position, just like junior, subjunior. It is below the terminal position, so we call it a subterminal in position. Or you can also say it as centromere is nearer to one end, one arm, which forms the very shorter arm. So nearer to one arm, forming a shorter arm, and the another longer arm. Okay, so acrocentric chromosome, centrosome, is close to one of its end, forming very short arm, and towards the other side, very long arm. So very short arm. So here it is shorter arm and longer arm. Here we call it as very short arm and very long arm. You can notice in acrocentric chromosome. A centromere is subterminal in position. Telocentric chromosome, the centromere is terminal in position. Okay, so these are the four types of chromosomes based on the presence of centromere. There are some chromosomes without centromere, we call it as acentric chromosomes. So chromosomes which do not have centromere, we call it as acentric chromosomes. Some of the chromosomes are having secondary constrictions which are non-staining, and this secondary constrictions we call it as satellite. Chromosomes which have the secondary constrictions or satellites, we call them as SAT chromosomes. Then organelles, you also find microbodies. What are microbodies? These are membrane bound minute vesicles that contain various enzymes. So they are present in both plant and animal cells. So microbodies are found, so which are membrane bound minute vesicles, microscopic vesicles. Already it is microscopic, but much more minute uh, vesicles which contain various enzymes. So microbodies are membrane-bound minute vesicles that contain various enzymes. So it is present in both plant and animal cells. So microbodies are present in both plant and animal cells. So they are membrane-bound minute vesicles which contain various enzymes. Microbodies are membrane-bound minute vesicles which contain various enzymes. So microbodies are present both in plant and animal cells. A few differences before we wind up the topic. A few differences between plant and animal cells is plant cell, they have cell wall is present, animal cell wall is absent. So plastids are present in plant cell, plastids are absent in animal cell. Plant cell has a large central vacuole, animal cell has many small vacuoles are there. So centrioles are absent in plant cell, animal cell, the centriole is present, which has the centrosomes or centrioles are present. So these are the differences between a few differences between plant cell and animal cell, and most of you have written much more exhaustive differences between plant cell and animal cell in the assignment that I had given. Okay. Then comparison between prokaryotic and eukaryotic cells. Prokaryotic cells generally they are smaller when compared to eukaryotic cells. Genetic material in prokaryotic cells is in the form of nucleoid or incipient nucleus or false nucleus. So whereas in eukaryotic cells, we call it as nucleus because they have a nuclear membrane and nucleolus. Whereas in case of prokaryotic cells, there is no nuclear envelope and there is no nucleolus. That is the reason we also call it as false nucleus or incipient nucleus. And this naked DNA material, we call it as nucleoid. So nuclear membrane is absent in prokaryotes. It is present in eukaryotes. Membrane bound organelles are absent in prokaryotic cells. They are present in eukaryotic cells. So the prokaryotic cells have circular DNA, whereas in eukaryotic cells, they have linear DNA. Ribosomes are of 70S type in prokaryotic cells. In eukaryotic cells, they are 80S type. Okay, 70S type of ribosomes are found in chloroplast and mitochondria. So these are the things that you have to remember regarding the differences between prokaryotic cells and eukaryotic cells. So you have listed out much more differences than what is given. See, prokaryotic cells, they would be undergoing amitotic cell divisions like binary fission and budding. Eukaryotic cells, apart from amitosis, they also exhibit mitosis and meiotic cell division. So they are all other points that we have listed out for prokaryotic cells and eukaryotic cells. So that completes our topic of cell, the unit of life. Okay, and we'll also be discussing about the MCQ questions, multiple choice questions in today's afternoon session class. We'll be discussing about this multiple choice question of cell, the unit of life. Okay, so that completes our topic for the cell, the unit of life. So we'll be starting with the next topic, that is cell cycle and cell division. That would be the next topic. As of now, have you understood this topic? Do you have any doubts or clarifications? You can get it clarified.